CRISPR sequences. What is mean by CRISPR? CRISPR is nothing but the clustered interspaced regulatory short palindromic sequences. That is palindromic repeat sequences. These are the important sequences now which is used for the development of various vaccines. It is nothing but a genome editing tool. What is mean by the genome editing? What is the necessity of the genome editing? That is, various applications are used for the genome editing tools. In the genome editing, it is used to study the various mutagenesis processes. Various mutations are studied by the genome editing tools. CRISPR is also used for the genome editing. It is used to study the site-specific mutagenesis which is occurring in the small interference RNA, that is the siRNA. Mutations or site-specific mutagenesis is studied in the siRNA, which is done by the CRISPR sequences or CRISPR sequence which is found in the microorganisms. CRISPR it consists of the CAS genes. Cas genes are present in the CRISPR sequences. It is used for the development of the adaptive immunity in the microorganisms. It was first demonstrated in the eukaryotic cell. The first CRISPR sequence was first demonstrated in the eukaryotic cell, whereas it is also used in the other systems also. When you look upon the evolution of the CRISPR, it was first started in 1979 and the research continued up to 2013. CRISPR technology. Okay. Next is the mode of action of the CRISPR sequences. In the mode of action, you have the three different types, that is the adaptation, expression and interference. Adaptation is first phase, next is the expression phase, the third phase is the interference phase. First, in the adaptation phase, the CRISPR is the locus undergoes the transcription and the transcript is processed during the expression phase, and during the interference phase the Cas protein or CAS protein is converted into RNA complex. CAS protein is converted to RNA complex during the interference phase. There are usually three different types of Cas proteins, that is the type 1, type 2 and the type 3 proteins based on the expression of the RNA. They have classified into three different categories. The protein which is associated with the CRISPR sequences is the Cas9 protein which contains the guide RNA. gRNA proteins, gDNA proteins are associated with the CRISPR sequences. It is involved in the type 2 CRISPR mechanism. Already I told you that it is responsible for determining the adaptive immunity. The first observed adaptive immunity in microorganism was first demonstrated in Streptococcus pyogenes. Streptococcus pyogenes, the adaptive immunity was first demonstrated. The basic structure of the CRISPR is it contains the six domains. The basic structure of the CRISPR is it contains the six domains: REC1, REC2, and other helix domains are found. REC1 is the responsible for it contains the guide RNA. It contains the guide RNA is present in REC1. In the REC2 protein is not very well studied. Apart from that, it consists of the PAM matrix, and other domains are also associated with the CRISPR sequences. The main structures are REC1, REC2, and bridge helix and PAM matrix. The three basic types of the CRISPR are wild type, Cas9, D10A, and dCas9. These are the three different basic types of the CRISPR sequences. And what are the mechanism? What is the main mechanism which is found in the CRISPR is the genome editing mechanism and genome engineering, which is done by the Cas9 nickase enzyme. Genome engineering is done by the Cas9 nickase enzyme. The third mechanism is the nuclease. Cas9 nuclease is an enzyme which is used for the another mechanism for the localization of the particular genome. In these three mechanisms, the particular protein, guide RNA protein, is activated and repressed. The last step, it is expressed in a particular sequence. That is, these are the three mechanisms, basic mechanisms of the CRISPR sequences. That is genome engineering by double nicking, that is by Cas9 nickase. The last mechanism is localization with detection of Cas9 nuclease. Cas9 nuclease is also involved in the another mechanism. In these three mechanisms, activation, repression, and expression of particular target specific proteins will take place. So what are the various components of the CRISPR techniques? That is the photospacer elements, CRISPR RNA, cRNA. Next is the trans-activating RNA, PAM, which I have already told you. Photospacer adjacent motif. Next is the CRISPR RNA trans-activating cRNA, that is called as the trans-activating RNA. There is in the adaptive immunity, particularly in bacteria, you have the two different types, that is the class 1 and the class 2. The class 1 proteins it uses the several Cas9 proteins along with the cRNA. In the class 2 proteins, you have a large component of single complex of Cas9 protein which is conjugated with the cRNA and the tracer RNA. Finally, we will discuss about the few applications. What are the different applications of the CRISPR sequences? That is the gene silencing, homology directed repair, and presently it is used for the development of various vaccines by the genome editing tools. Thanks for watching. Thank you.